DNA test for growth, feed efficiency, carcass and reproductive performance. We're lucky tonight to be joined by Mike Goddard, who will be presenting the majority of this webinar. Mike is a professional fellow in animal genetics from the Melbourne University, but most importantly in this area he was the Beef CRC chief scientist. So he's got very much a solid background, and a lot of the work that's gone into this area has been led by Mike. My name's Christian Duff. I'm with SBTS, and I would know most of you, and I'll be facilitating this webinar tonight. Okay, just to let you know, this is the webinar series, The Cutting Edge, and the reason for doing these sessions is obviously the Beef CRC closed its door on the 30th of June this year, but there's obviously a lot of really important research to get through, and there's a lot of good research outcomes coming out after the close of the CRC, which researchers such as Mike are working on. So it's pretty important to get these messages out. This is one way we can do it, and tonight's webinar, as I said, is on the Beef CRC DNA test for growth, feed efficiency, carcass and reproductive performance. If you're interested in any of those other sessions we've done, the previous four, and you haven't been to see them, we've actually recorded those, and you can get those on the SBTS or TBTS websites under the webinars page. And if you just go to past webinars, they're all recorded and actually recorded and loaded up to YouTube. So that'll allow you to see those, and we encourage you to look at those if you haven't seen them. So that's what we're up to now. What I'm going to do now is I'm actually going to take a second and hand over to Mike, who's the presenting. I'll put him back on, unmute him, and I'll hand the presenting over to him, and you'll be able to see his presentation, which we've outlined. Okay, Mike, so we can see your presentation.
Hello, everybody. I'm just trying to get this to full screen. So this is about the Beef CRC hopefully delivering genomic predictions for breed plan. For some reason my slides aren't moving. So I'll start with the take-home message. The Beef CRC has developed prediction equations to predict breeding values from DNA markers, and these are prediction equations are being transferred to breed plan and genomics companies, and the product for cattle breeders is more accurate EBVs, which incorporate DNA information. So some background. Some traits are due to a single gene, such as red versus black coat colour or horns versus poles, but most important traits are not like this. They're quantitative or complex traits, and they're controlled by many genes and environmental factors, and growth, fertility and carcass composition are all like this. Traditionally, we improve these traits by selection on performance and pedigree using EBVs. So we've got two sources of information, performance and pedigrees. We put them through a computer and we come out with pre-plan EBVs. And this works, but it's slow for some traits. Traits like cow fertility, where you can't evaluate the bull without doing a progeny test, are harder to improve with traditional means than something like weaning weight, where you can easily measure both bulls and heifers. Beef quality is another problem. If you've got to kill the animal in order to evaluate it, it's a bit late then to decide to choose it for breeding. However, a remarkable thing is that the genetic variation in all of these traits are due to differences in DNA sequence. DNA sequence is just a long list of 4 different letters or bases, C, G, T and A, in all sorts of different orders. Here's just a little bit of a sequence, CG, C, T, A, T, T, G, and here's the same sequence from another animal, and it's got an A where the first one had a T. And it's these differences in the sequence that cause different genetic differences between animals in everything. This particular sort of difference in sequence is called a single nucleotide polymorphism because it's just a change in a single letter. And single nucleotide polymorphism is abbreviated SNP, or SNP for short. So you'll hear me and other people talking about SNPs. They're just places in the DNA where there's a variation in a single letter. So an obvious suggestion is to find the DNA sequence variants that alter important traits and to select for the version that gives the most profitable outcome. The trouble is we can't do that. And the reason we can't do it is that we don't know which SNPs actually alter the performance of the animal. However, what we do have is lots of random SNPs scattered all over the genome. And there's so many of them that it means that any gene which actually causes variation in a trait has got to be near to one of these SNPs. And so we can use the SNPs to sort of track the inheritance of the genes that really matter. Here's an example where we've got one marker, it's a CG SNP, and we've got a gene that affects a trait. Let's say it's marbling. The plus allele increases marbling, the minus allele decreases marbling. And in this hypothetical example, if you've got the plus allele for marbling, you've usually got the C at the marker, at the SNP. And if you've got the minus allele for marbling, you've usually got the G at the marker. The other ones occur, but they're rare. So consequently, if you select animals carrying C, most of the time you get the positive allele for marbling. So we've used hundreds of thousands of SNPs scattered all over the genome to track the causal variants, the changes in DNA sequence that actually cause differences in performance, and we use them to predict the breeding value for economic traits. This is called genomic selection, and it's being used by the dairy industry worldwide and increasingly by other livestock and even crops like wheat and maize. Its benefit in livestock is greatest for traits that are hard to improve by traditional means. And I've got a list here: carcass and meat quality, food conversion efficiency because it's very expensive to measure, adaptation to the tropics, especially if you don't live in the tropics, fertility, as I've already said about cow fertility, and polled because an animal can be polled but carrying the horn gene. So we use the DNA data, the data on the SNPs, as an extra source of information to calculate EBVs. The aim is still to get the most accurate EBVs for cattle that we can, but as well as the performance, the phenotype and the pedigree that we've always had, we've now got an extra source of information, which is the DNA genotypes at all these SNPs. So the aim of the CRC's program in this area has been to find genetic markers for these important traits, and the path to market is that a DNA service company types the markers, and then that information is used in the calculation of EBVs. So in this presentation I'll talk about how we do that, what the results are, the commercialisation of it, and the future. I might just stop there to see if there's any questions on that background material.
Hi Mike, there's no questions at this stage, and just to let you know, because when we're both on the line there's a bad echo, so I've now muted you and unmuted me. But so what we'll do is we'll pull up from time to time and ask questions. One thing I forgot to do at the start is just tell everyone that they can actually ask questions. You can't actually ask verbally Mike questions, but there's in your control panel you'll see in front of you there's a question submission field. So all you need to do is type your question in, press submit. I'll monitor the questions and we'll pull up from time to time and I'll ask Mike the questions on behalf of you and Mike will answer back. Just be aware though, I will be muting and unmuting Mike and myself just so we don't have that echo issue. But at this stage, Mike, there's no questions, so we can continue on to your next stage of your presentation. You're now unmuted.
Okay, so we'll keep going. The research strategy is really very straightforward. We have a discovery or training population, and they're cattle where we've genotyped them with the SNP chip, in other words, genotyped them for all these SNPs, and we've recorded or measured all of the traits that we're trying to predict. And from that data, we come up with a prediction equation that takes the genotypes of the SNPs and predicts the performance for the traits we're interested in. And then we have what we call a validation population, which is a separate group of cattle, and we test the prediction in the validation population. They weren't used in the discovery, but we use them by applying the prediction equation, predicting a performance, and then seeing if that's held up by the real performance of those animals in the validation population. The number of cattle that we had is shown here. We had a whole series of different SNP chips. The 700K means that it's a SNP panel with 700,000 SNPs on it. Similarly, 50K has got 50,000 and so on. And we did 1,698 animals with the very high density panel, the 700K panel, and then we did a larger number with the cheaper 50K panel. Towards the end of the CRC, the 7K panel became available and it was cheaper still, and we used it. And right at the beginning of the CRC, there was a 10K panel that we used for a small number of animals. So we ended up with about 14,000 animals genotyped. They were spread over 12 different breeds. There's Taurus breeds like Angus, there's Brahmins, and there's the composite breeds like Santa Gertrudis, Droughtmaster, and the tropical composites are from the pastoral companies in northern Australia. So pretty good range of breeds. Lack of Euros though. Unfortunately, we couldn't measure all animals for all traits. If you're going to measure tenderness, you have to kill the animal, and then you can't measure, if she's a cow, her lifetime fertility. So the number of animals measured for different traits varied, but it's about 4,000 or more for most traits, except bull fertility, where we only ever had 1,100 done for that. And what we do is that we impute 700,000 genotypes for all animals. So we've got the animals that were actually done with the 700K SNP chip, and we predict for all of the animals done with 50 and 7K what they would have had if we'd done 700K SNPs for all of them. And we can test whether this works or not by putting aside a group of animals that actually do have 700,000 SNP genotypes and seeing if they're predicted or imputed correctly, and we found an agreement of about 90%. So it wasn't perfect, but it was worthwhile saving the cost on the SNP chips by doing the 50 and the 7K and then imputing up to 700K. This just shows a little bit of information about the relationship between the different breeds. You can see over on the left-hand side there's a big cloud of green spots. Each spot is a Brahman animal, bull or steer or cow. So this this shows the relationships amongst the breeds as worked out by the SNP genotypes. And the Bos Indicus Brahmins are on the left-hand side, and over on the right-hand side are the Bos Taurus breeds. And they're sort of spread out in the second dimension from top to bottom, so that I think we've got Angus up the top, the purple ones, and then Murray Grey very close to them, and then some Herefords, and then down at the bottom some Shorthorns. And in between we've got the Brahman composite breeds, Santa Gertrudis right down the bottom, and then Drought Masters in a yellowy orangey colour, tropical composites in a light blue, and Belmont Reds in a very pale pinky colour. And it shows that the Belmont Reds are actually very like the tropical composites that the pastoral companies have got, at least on these two axes, which is just a sort of a first approximation to how the breeds are related to one another. So the SNPs can distinguish the breeds. More importantly, can the SNPs find genes for important traits? So we looked for a couple of genes that we know are there anyway. So they're a sort of a test. Can we actually find something that we know is there? This is a graph of results. Stretched out along the bottom is one chromosome. It happens to be chromosome 7. Cattle have got 30 pairs of chromosomes. This is chromosome 7. And the vertical scale says how strong is the association between one of the SNPs and the trait that we're studying. In this case, the trait is tenderness of the meat. And each blue dot is one SNP. So you can see that even on this one chromosome there's lots and lots of SNPs. But there's a very strong association just at this point here on the chromosome, and that's where the gene kelpostatin is. So we've found, using our SNPs, the kelpostatin gene that was already known to have an effect on tenderness. So that's a test to see whether the method works for finding genes that we know are there. This is another example of the same thing. This is chromosome 14 laid out from end to end on the bottom, and again up the vertical axis is how strong an association between each of the SNPs, and in this case it's hip height, frame score you might call it. And there's a big peak just here, and that's where there's a gene called PLAG1, and we already know from other work that PLAG1 affects hip height. So we're finding the genes that we know should be there. PLAG1 not only affects hip height, it affects a whole bunch of other traits, including growth and fertility and carcass traits. So we looked to see whether there were other genes that weren't previously discovered that affect many traits. And yes, we can find new genes. Here's chromosome 5, and there's a big peak on a gene called HMGA2, which doesn't mean anything to anybody, but it's a gene which is known in other species to affect growth and fatness. And lo and behold, in cattle, it does that. It one of the alleles, the other allele of course does the reverse of this, one of the alleles decreases height, decreases weight, decreases fatness at the P8 site, but increases fatness at the rib site, and increases marbling, increases eye muscle area, increases IGF level, decreases daily food intake, but not enough to compensate for the lack of growth, and so it increases net food intake. So this is a gene with a whole bunch of effects on growth, and importantly, it's redistributing fat from P8 to rib and marbling. In mice, if you knock out this gene, you reduce growth, especially fatness. And in humans, it changes the pattern of fat between putting it on around your belly and putting it on around your hips. So it's a gene that has effects on growth and fatness in humans, mice, and cattle. It turns out that the frequency of that allele is virtually 0 in the Brahmins and virtually 100% in the Bos Taurus breeds. So we only see it segregating, we only see it causing effects in the tropical composites. So you can, in the tropical composites, you can select for one allele or the other, depending on whether you want smaller cattle or bigger cattle, and with all the other things that go with that in this gene's case. But in the Brahmins and in the British breeds, you're virtually stuck with opposite alleles. However, that's not the usual situation. The usual situation is that these genes will either segregate in Brahmins, they'll be variable in Brahmins and fixed in Bos Taurus, or the reverse. They'll be variable in Bos Taurus and fixed for one of the alleles in Brahmins. So can we find the genes? Yes, we can, but the trouble is there's just too many of them. Here's a graph like the other ones, except all of the chromosomes have been put side by side, one in red and then one in black and then one in red and so on. And for this is for weight, and there's just hundreds of genes, probably thousands of genes that affect weight, and there's in fact hundreds or thousands of genes that affect everything we've looked at. So what we have to do is come up with a prediction equation based on all of the SNPs, using all 700,000 SNPs, rather than try and just find a couple of genes like PLAG and kelpostatin. We've got a single prediction equation for all breeds. That's an advantage because it means that all breeds are benefiting from the data set we've got, which is spread across 12 different breeds. And then we validate the prediction by testing in another group of cattle, and in order to avoid having close relatives in the validation data set, we make sure that none of the animals in the validation set have got the same sire as the ones in the discovery set where the prediction equations came from. And to evaluate how good the prediction is, we calculate the accuracy of the prediction equation. That's the correlation between the predicted breeding value using the prediction equation and the true breeding value. The same definition of accuracy as used by BreedPlan. And here are the accuracies for the different traits that we looked at. They're not brilliantly high. They're certainly not 100% accurate, but they're high enough in many cases to be useful. Feed conversion efficiency 0.44 is good because it's very hard to improve food conversion efficiency otherwise, because it's just too expensive to measure food intake. Tenderness 0.38 is good because it's hard to measure tenderness on live animals, or in fact you can't. Retail beef yield 0.13 is pretty disappointing. So some traits were easier to predict than others, but in general we've got predictions which are of some value to cattle breeders. They vary a little bit between breeds. Here I've got 4 breeds just as examples, and 2 traits, tenderness and weight. And in general, we did better in the tropical composites, the Brahman composite breeds, than we did in Bos Taurus breeds like Angus and Hereford. We tried using a prediction equation where we only used the same breed for discovery as we used for validation. So we worked out a prediction equation for Angus using only Angus data, and that's what I called here the within breed accuracy, as opposed to using all the breeds, which is what I've given you previously. And I've done it here for weight and for net food intake, and most of the time the all breeds prediction is better than the within breed prediction. So we think it's worthwhile using all of the different breeds to work out a single prediction equation, and then using that for each breed that needs to make a selection decision. However, this is still an area of research to see what's the best way to do this. So we've done validation with the cattle that we've got already, but there'll be future validation done by ABRI and by other people, and they've got some industry sires, some young cattle, the bins or beef information nuclei that many breed societies have set up, and we have some Canadian cattle, and we could even use dairy cattle if there's a trait that's measured in both beef and dairy. ABRI are in the process of doing this, and I think we'll have to wait for a little while until we've got all the answers from this additional validation. But the best numbers I can give you at the moment are the ones from the validation the CRC did, which is what I've put up till now. I'll just stop there again and see if there's any questions.
Thanks, Mike. Yes, there are a few questions here, and they're all similar, so I'll word them all in one question, then I'll hand back over to you. I guess it comes down to we've seen what the CRC prediction equations are trained against in regards to the breeds in the population. How will this affect the accuracy? For example, there's a quite heavy bias in the population towards Brahman and tropical composites. How will this affect the accuracy of the test for British breeds or European breeds where the numbers were quite low in the training and validation population? Can you just comment on that in a general terms? I'll just switch it back on. Okay.
It does require animals within the breed where you want to use it. So I wouldn't have any confidence of these prediction equations in Wagyu because there's absolutely no Wagyu data in the discovery set. For Angus, there's quite a decent amount of data. For European breeds like Charolais and Limousin, there's much less. And so, to be honest, I wouldn't expect it to work as well in breeds like Limousin where we don't have as much data as we have for Angus. But we'll really only know that as more work is done to check this out. And really what we need for any breed that wants to use these predictions, we need to have some animals from that breed in the discovery set so that the prediction equations take into account what happens in that breed.
Thanks, Mike. I'll probably just add there, in the industry size and industry young animals, there is quite a good representation of European breeds. So even though the Europeans didn't have a high reference in the training population, at least they've got animals genotyped that will be able to, ABRI particularly, be able to do some research to see how well do the CRC prediction equations work for the European breeds as such, and for other breeds that have done that testing. So I think that's a good place. Do you want to add to that, Mike?
Yes, I think there's two stages to what you've just described. One is using the industry size and young animals in, say, Charolais or Limousin to test the existing prediction equations, but the other step is to add that data to the discovery set and come up with better prediction equations. Of course, we still need to leave some animals out so that we've got a validation group that's independent. But I'm sure we could do better for Limousins and other breeds by using some of these new animals, either from the bins or from the industry size or industry young animals. Using some of them will help us, I think, in getting better predictions for breeds that were poorly represented in the CRC's own work.
Thanks, Mike. Just another question here before we move on. What's actually happening in overseas countries? Are they going down the same prediction equation path across breeds and then applying it to individual breeds? Or can you just give an insight on what you're seeing happening in overseas areas where they might have bigger populations that have been genotyped? Over to you.
To the extent that I'm aware of this, and you know, it's not, there may be a lot of places I'm not sure what's happening. In the US, the Angus have got their own prediction equations, and I think breeds like Limousin, which have genotyped a lot of animals, will also have their own prediction equations. Our assessment is that this is not the best way to go. We get more accuracy by using all of the cattle rather than just the ones from the target breed. But in other countries where this might be controlled by a breed society, for example, it's harder for them to combine the data across breeds. The crossbreed advantage is particularly acute in the composite breeds like Santa Gertrudis, Droughtmaster, Belmont Red, and the industry tropical composites, because for those breeds they've got animals that have got relatives that are Brahmins and relatives that are Shorthorns or Angus or something else, and so they particularly benefit from being able to put all of the cattle together. But I believe all of the breeds will benefit in the long term from having a combined discovery set. And one of the reasons for this is that it'll produce more stable prediction equations. The trouble with the prediction equations, if you say only use Angus for your discovery set, is that while it might work in your Angus, it doesn't work so well in somebody else's Angus. And maybe it doesn't work in the Angus of the next generation or in 5 years' time. Whereas if you have something that works in Angus and Hereford and Shorthorn, it's probably going to work in next year's Angus as well. So I think the crossbreed one, in my view, is the way to go, but it's not what's being done in most cases overseas that I'm aware of.
Thanks, Mike. Some more questions come in, but what I think we might do, we might go on to your next part of your presentation, and then we'll pull up for another question session after that. So back over to you.
Okay, well, let's go on. What's the commercialisation of these prediction equations? The outcome that we want is breed plan EBVs that incorporate the DNA data and are as accurate as possible. Why is that the way to go? Well, it really isn't helpful in making selection decisions to have lots of different independent bits of data which are telling you different things. You know, you might have a young bull whose sire is terrific, whose mother was a bit ordinary, whose own performance is great, but whose sister is pretty ordinary. Now, breed plan EBVs put all that information together to give you the best estimate of the breeding value of the animal. And we want them to do the same with the DNA data. We want them to use all the available data, including the performance and pedigree data, plus the DNA, to come up with the best EBV that we can. So that's what we're aiming for. We've got two models for how to get there. The one is that the genomics company genotypes the cattle. I've called them selection candidates because they're the animals you're trying to make a decision on. The genomics company applies its own prediction equation and gets what I call a marker breeding value. In other words, an EBV that only comes from the marker data. And they send that to breed plan, and that marker breeding value, or MBV, gets incorporated into the breed plan EBV. That's one model. The second model is that the genomics company genotypes the cattle, the selection candidates, and sends the raw genotypes to breed plan, who apply their own prediction equation, for example the CRC one, to get their own MBV, which gets incorporated into the breed plan EBVs. So the CRC's policy has been to support both of these models. We will give our prediction equations to companies like Pfizer and Merial, and we will also give our prediction equations to breed plan. If you want my personal opinion, the second model is the better one, because breed plan then has access to the actual genotypes for all the SNPs, and for lots of reasons that's a better way to go. But we've said as the CRC that we would provide our information to both breed plan and to the genomics companies if they're offering the service in Australia. That's what this says. We give our prediction equations to both. How does a cattle breeder actually use it? It's easy. You take a sample which can provide DNA, like a tail hair root. You send that to the lab. They genotype the animals, and the prediction from the genotypes goes into breed plan. So how do you use the outcome? Well, really, you select on EBVs just like you always did. It really doesn't change the way in which you would select cattle. What it does give you is an opportunity to get more accurate EBVs and therefore make more progress for fertility, feed conversion efficiency, tenderness, and traits like marbling that are, you can measure them on the live animal, but it's difficult. Is it of value to industry? These are some back-of-the-envelope calculations that say if we could increase the accuracy with which we select animals for dollars from 0.3 at present to 0.4, then that'd be worth millions of dollars. Now, you're probably all very skeptical of these sort of calculations, so I'll just say that with something as big as the Australian beef industry, even a rather modest improvement is worth a lot of money globally. What about, though, to an individual stud breeder who's got to pay for it? Well, we get slightly more accurate EBVs for traits like food conversion efficiency, meat quality, carcass, and fertility. How would a stud breeder use that? By retaining the best bulls for use on the stud and selling bulls with more accurate EBVs and making more accurate decisions to cull the worst of them. Now, the value of that depends on a lot of assumptions, like what trait you're selecting for, but we think it's at the order of $100 to $600 per bull tested. So provided you can get this service for less than $100, it's probably worth doing. The caveat is that you have to be able to get a higher price for better bulls, because what this offers you is a way to breed bulls with better EBVs and sell them for a little bit more money. If you can't sell better EBVs for more money, well then of course it's hard to justify paying for the cost of the DNA test. Future research, this is after the CRC, so with no guarantee that any of this will happen, but the sensible thing to do is to try and increase the accuracy of the EBVs. I gave you figures that were 0.4 roughly, or less, 0.4 at least by the time you incorporate the traditional information, but what we'd like to do is push that up to 0.6. That would be more useful to cattle breeders, and we've got a whole lot of research ideas as to how we could do that. And I won't bore you with the detail, but this is a field of research which is really only just getting going, and so I'd be very surprised if we can't improve upon the modest success that we've had so far. The best evidence that you can get it to work better comes from the dairy industry, where they've already got accuracies of over 0.7, 0.7 and 0.8. So you certainly can make it work. The reason that it works in dairying is that they've got very large populations, all within Holstein, and all accurately assessed by progeny test. The US, for example, has got a discovery population of 14,000 progeny-tested Holstein bulls. So you can make it work if you've got good enough data. And so one of the things we have to do in beef is to get better data. But we're never going to have the quality of data across all of our beef breeds that they've got in Holstein. So we've got to do smarter things, and that's why I've got this list of ideas for research, and one of them is to use the actual DNA sequence data, where you don't just genotype them for 700,000 SNPs, you actually sequence the whole genome of the animal so that you find the mutations that actually cause changes in the performance of the animal, rather than just these markers that are sort of nearby, which are imperfect guides. If we can get the actual genes that cause variation, then we'll get better, more accurate predictions. So conclusions, the CRC has delivered prediction equations to breed plan. They'll be incorporated into EBVs, and that'll give us a small increase in the accuracy for hard to measure traits. And there's a whole bunch of people who've contributed to this, and going back over 18 years that the Beef CRC has been collecting data. So I'll stop there and answer questions if I can.
Thank you, Mike. Thank you for taking us through that information. We do have a range of questions here which we'll go through now before presenting back to me to finish off for the night. Quite a few around the commercialisation model that you went through. You had two options there. One is the MBVs coming straight back from the genomics lab. The second is the genomic results coming back and the prediction equation applied via breed plan. The question here is quite specific, but I'll read through it. Under the commercialisation in Model 1, can the beef CRC predictions be blended with existing predictions that have been provided by other commercial providers? So as you know, there's some MBV genomic information coming into some of our analyses now for breed plan. How can and will it be able to be blended in with the beef CRC information and vice versa? Can you just comment on that?
There's a couple of different possibilities. None of them are in operation today, but we are giving our prediction equations to companies like Pfizer and Merial. What they choose to do with them is up to them, but one of the things they could do is to combine the prediction equation with their own data and come up with a better prediction. The clearest case of that is where they don't have any data of their own at all. For example, for fertility in Northern Australia, then they might just use the CRC prediction. But for traits where they've already got their own prediction equation, like for marbling in Angus, they could combine ours with theirs and hopefully produce a better prediction equation. So that's one possibility. Another one is for a company like Merial, or they're now got merged with GeneSeek, and I've forgotten the name of the new company. They might agree to put into breed plan both their MBV and the raw genotypes. That would allow breed plan to use both their MBV and its own internal prediction equation. So that's another possible way in which the combination could happen. The third possible way is not a desirable one. The third possible way is that you send your DNA sample to a company who calculates an MBV, and you also send it to a lab that does the genotyping and puts the data straight in. Of course, that allows breed plan to use both sources of data, but of course you have to pay twice. So we need to work out a way of avoiding that, where you only pay once, but you can get access to both the MBV from the company and the raw genotypes. In the past, Pfizer have been unwilling to put the raw genotypes into breed plan. Whether they would have a change of heart in the future, I don't know. I think Merial is more likely to agree to put the raw genotypes into breed plan.
Thank you, Mike, for answering that. Just another couple of questions around the area of commercialising this test, and I guess it's more focused on the commercial industry rather than the C-stock industry. Obviously, with the preferred model, the information will be flowing through breed plan, but then you have to be, I guess, a herd recording with breed plan. What are your thoughts about offering DNA test and prediction equation results that any commercial producer can get access to then maybe test their cow herd to get some pitch on their genetic value of their cow herd to breed from there? Is that a viable option, or what are your thoughts in that area?
It's a possibility. Technically, there's no reason why it couldn't be done. If you had a commercial cow herd, you could take DNA samples from them, get them genotyped, run them through the prediction equation, and get an EBV or an MBV for your cow herd. What would you do with it once you'd got it? You know, you've paid a fair bit of money for this, and if you're a commercial producer who buys bulls, I'm just not quite sure what you would use it for. if on the other hand you're a large commercial producer who breeds some of your own bulls, then you could use it, you could genotype your bull calves, not the cows, and make a selection decision amongst the bulls. But then you're behaving exactly like a stud. You know, you're running your own stud, and you'd be well advised to put the data through breed plan in order to get the best EBVs you could. So I'd be happy for people to genotype their cows, but I'm very reluctant to advise it because I just can't see that they can get enough economic benefit from it to justify the cost.
Thanks, Mike. Just another, I guess this is probably more of a comment, but it'd be good to get your feedback on it. Obviously, seed stock producers now collect DNA for parentage verification, genetic conditions, horn polled in some circumstances, and now again for inclusion of this information through genomic predictions into EBVs. Could you just want to comment on what you see the progression towards just one simple genomic test to cover all those areas? Is that going to be possible? Do we know it's coming? What are your comments there?
Okay, so this is, of course, exactly the right way to go, and it is coming. The company that's been doing genotyping, GeneSeek, has merged with Muriel, and unfortunately I've forgotten the name of the merged company.
Might be Neogen.
They're developing. Sorry?
Neogen, I think, is the...
Neogen, of course. So Neogen is developing a 10,000 SNP chip or panel, and it will have on it all of the SNPs for parentage and also for known recessive abnormalities where they've negotiated with the patent holder to have the right to genotype this particular abnormality. So I think we're very close to having a very useful DNA test out of Neogen that will have the about 7,000 random SNPs that can be used for the prediction of breeding value that I've described, plus parentage, plus known abnormalities.
Thanks, Mike. Just a simple question for you now. In one of your slides, you talked about a prediction accuracy, a genomic prediction accuracy of 0.6. Do you mean this is related to a breed plan EBV accuracy of 60%?
Yes, that's correct. I probably should have said 60%, but it's the same meaning of accuracy as breed plan uses.
Thanks, Mike. Now just a couple of questions, probably just to finish off the questions for the night. Pretty much related to specific breeds, but I'll word them the same way. I guess the thing is, I have to, based on this, it'd be good to get some DNA testing done, or I've submitted samples for the young animal project to get some genomic results and help out with the validation work that ABRI is doing. Do we know yet when that will be incorporated into breed plans, or it's still something that's in a work in progress and something we'll know about soon? I guess the timeline is people thinking about, and will it apply to my breed? When will we know those good hard facts?
I can't fully answer that. I don't have any control over the decisions made by breed plan. I know that the scientists at AGBU are working as hard as they can on the validation and the incorporation of these prediction equations. But when you're doing something completely new, to be perfectly honest, it's very seldom that it works the first time. And so I think we're going to have a little bit of a delay while everybody's check that we've got this right, rather than sort of rush in order to say we've delivered and later have discovered that there were mistakes made. So I understand people's impatience. We've been promising this for a long time, but it's turned out to be technically harder than we thought, and I think we should still be a little bit patient until we're sure what the answers for your particular breed are.
Thank you, Mike. We might pull up the questions there for the evening, and I just want to thank you for your presentation. But I'll now just hand over presenting to me, Mike, and I'll just finish up. So if you can stay online, we'll just finish up the presentation. If everyone else can stay online too, just because I've got a few things I need you to do for me. So just bear with me while I change presenters, and I'll show my screen, which you'll see there now. The first thing I want to do tonight is to do a poll, and the first poll will come up in front of you now. I'll just launch that poll, and if you can all take a second or two to answer that. The first poll, as you can see there, is: did you find this webinar useful? Strongly agree, agree, unsure, disagree, and strongly disagree. If you can just spend some time answering that, and I can monitor what percentage of votes have come in. We've got about 62% voted. Let's try and get up to about 80% would be good. Okay, I'll close that poll off in a second, and I'll launch a second poll. So I've just closed that poll, and I'll now launch a second poll for you to answer. Basically, this is saying, based on this webinar, are you more informed about the application of the outcomes from the Beef CRC within your enterprise? And again, strongly agree, agree, unsure or not applicable, disagree or strongly disagree. Again, thanks for putting a bit of effort in answering these. It's good for our feedback. Okay. I'll just close that off. Thank you for your time, and back to the presentation. So there are the polls. Basically, as you know, we've now completed this webinar, and these webinars will be made available from the SBTS and TBDS website. We basically record them and we can upload them to YouTube. And basically, if you just go to the SBTS and TBDS website, you jump on a YouTube click and you can see the presentation like you've done tonight and the previous webinars. If you'd like the webinar course, that's the 6 webinars which we will put in together after the next week's one. You can let us know and we can actually get you on CD if that's a preferable way of looking at it, if you've got bandwidth issues or something. And most importantly, you can also answer or access answers to any of your unanswered questions by contacting the allocated technical officer of your breed. And I'll put those up there now. There's Paul Williams, Andrew Byrne, myself, Gemma Wilkinson, Peter Parnell and Alex McDonald, and the applicable breeds next to those. So I want to thank you for participating in this webinar, but most of all, I really want to thank Mike Goddard for putting the effort in tonight, giving us some good information out of the CRC, and no doubt we'll hear much more in the short term about how this information is going to be applied through breed plan. So thank you, Mike, and thank you all for logging in tonight, and we look forward to seeing you next week's webinar on the poll gene. Good night, everyone.